Hi, my name is Atefeh and I'm a postdoctoral researcher in Dr. Riang's lab at Hotchkiss Brain Institute at the University of Calgary. And today, as a part of Fundamental Discovery Week, I would like to explain how basic science helps us to make new discoveries in multiple sclerosis research. My journey into this field began when I was working as a nurse in an intensive care unit of hospital. At that time, working with individuals with neurological disorders made me curious about how brain works and what's the underlying mechanisms of neurodegenerative disease. That was why I pursued graduate studies in neurophysiology. In 2017, I joined Dr. Javan's lab at Tarbiyat Modares University, Tehran, as a PhD student, and in his lab, I worked on the mechanisms of neurodegeneration, the cellular and molecular mechanisms in the context of multiple sclerosis. But when it came time to choose a postdoctoral position, I wanted to be in an environment that not only discovers mechanisms of the disease, but also pushes discoveries toward treatment. And Dr. Riang's lab at the University of Calgary exactly offered that. In his lab, fundamental research and therapeutic goals are tightly connected. So I joined Dr. Riang's lab in June 2023, and since then I'm working on multiple sclerosis. In MS, immune system attacks protective myelin around nerve fibers, disrupting the communication between cells. When myelin is damaged, the neurons can no longer send signals efficiently, and this significantly slows information flow, which affects memory, sensation, movements, and many other functions. At the same time, the immune response becomes amplified, and several immune cells from periphery enter the brain and the spinal cord, and together with resident microglia, they produce an inflammatory environment which is not permissive for myelin repair. Myelin cells, including microglia and macrophages, release several harmful molecules, including reactive oxygen species. But our cells have defensive mechanisms that can detoxify these reactive species. However, when there is an imbalance between production of antioxidant, oxidative stress happens. Currently, we don't fully understand oxidative stress in MS and how we can protect against it, which makes developing treatment challenging. In this project, I focus on an enzyme which is called NOX2 and is highly expressed in myelin cells, in microglia and macrophages, and my research has shown widespread expression of NOX2 within MS lesions. So we're trying to understand how NOX2-driven oxidative stress can injure neurons and also inhibit myelin repair. Also, we're interested to see whether NOX2 deficiency and inhibiting NOX2 activity can lead to can enhance myelin repair and also protect neurons. So in this project, I'm using genetic tools to delete NOX2 from mouse genome. And so far, my research has shown that NOX2 deficiency reduce the level of oxidative stress and also the extent of demyelination in animal model of MS. Also, in collaboration with Dr. Cecil Lick, who is the chemist at the University of Calgary, I'm testing novel antioxidants to see if they can protect neurons and also enhance repair. Overall, this project will advance our understanding of oxidative stress in multiple sclerosis at basic level.